Hi, welcome to the Bioinformatics Chat. My guest today is Hamish King. Hamish is a postdoctoral fellow at Queen Mary University of London, and we will be talking about immunology and specifically about a study that Hamish and his co-authors recently published in Science Immunology, and that is about B cells and B cell states and B cell receptors. A lot of interesting stuff there. So, Hamish, welcome to the podcast.
Thanks so much. It's great to be here.
Just to not scare people away, especially those who don't routinely study immunology, let's do a brief recap of the main immunology concepts. So maybe let's do a quick introduction to the B cells and B cell states and their life cycle.
Yeah, that's a good idea. B cells 101. And I should maybe start by saying that I'm only recently arrived in the world of immunology, that my background was in sort of chromatin genomics and epigenetics before this. So I found it similarly overwhelming when I started into the world of immunology. But I think the major things to think about B cells, I guess, are why they're important, first of all. So the major role that B cells play in our immune system is by recognizing foreign pathogens and making antibodies to fight them off. And these are one of the major ways that our bodies recognize infection and fight infection and remember it as well, because some of these B cells are called memory B cells or plasma cells. They're two different lineages that can form, hang around in the body for up to decades after you might get sick, and can be reactivated when you get exposed to a virus or a bacteria again. So this is, I guess, why it's really important to understand how B cells are made, and in particular, I'm interested in the genes that control that process and control the selection of these B cells to make high quality and high affinity antibodies. So I think that's us into why. I think it's also worth touching on how this happens, because this is one of the main things that we try and address in our study. And there are two main stages of B cell development that are important to sort of separate. And the first occurs in the bone marrow, where we go from a hematopoietic stem cell through the B cell commitment to form a mature, naive B cell. So that's a little bit of a contradiction in terms, perhaps. So a naive B cell is a B cell that hasn't been exposed to any antigen yet, because it's just been derived from the bone marrow. And during that process in the bone marrow, they're making a first draft of the antibodies that they can make. So the antibody genes, which are sort of all exist in one locus in the genome with the variable diversity genes, undergo a recombination process which generates a huge amount of diversity before the B cell has even seen an antigen from outside the body. And this can generate, I think, estimates up to 10 to the power of 14 different unique antibody sequences. So when it exits the bone marrow for the first time, this naive B cell is able to make an antibody, but it's fairly non-specific. It won't do a great job of fighting off any infection, because that B cell hasn't seen anything yet. But it has a highly complex background none the same. So from there, the naive B cell is moving through the body in the blood or the lymphoid system, and is passing through secondary lymphoid organs like lymph nodes, the spleen, or the tonsil, which is our system of choice for our study. And when they're passing through these systems, they can be presented with a foreign agent. So this could be part of a virus, it could be part of a bacteria, and the B cells are presented with that with some help from T cells and other immune cells like a macrophage, and they sort of get activated and a signal to undergo this maturation response, and they form something called a germinal center. And in the germinal center, the B cells will divide rapidly and clonally expand. And as they undergo that clonal expansion, they can acquire mutations to their antibody genes. And sometimes those mutations will be beneficial and will increase the affinity of that antibody to the foreign agent. And if that happens, the B cells will get a strong signal to proliferate and strong survival signals. On the other hand, if they get a mutation that introduces a stop codon or a frame shift or other deleterious effects into the antibody genes, they won't receive those same signals and they will apoptose and die. So this is a really important selection process through which the B cells acquire a high affinity towards their target that they've recognized in some way initially. And then from there, this can happen many times. It's sort of a very iterative process where the B cells are cycling back and forth between different parts of the germinal center, which are these histological structures in the tissue. And at some point, they can differentiate from that germinal center and exit to form these memory B cells or plasma cells. And that is when they go out and do their thing and sort of suppressing the infection. And so does that sort of catch you up, Roman? Did that all make sense?
Absolutely, yes. I think that was very helpful. And maybe one more concept that is good to understand in order to better understand and appreciate your paper is something called antibody class or an isotype. So how does that work?
That's a really important point. I can't believe I forgot to mention that. So there's two main parts of selection that these B cells are going for relating to the antibody genes in the germinal center. One, which I already talked a little bit about, is these mutations that occur to increase the affinity. And this is a process called somatic hypermutation. The other really important feature of the antibody repertoire is the class switch recombination. So when the B cells exit the bone marrow, they will all make the antibody isotype IgM. IgM and IgD are typically what naive B cells will make. When they go through the germinal center, though, they can switch their antibody class from IgM to another class of antibody. And broadly speaking, it would be IgG, IgA, or IgE. And then there are subclasses as well, IgG 1, 2, 3, and 4, and IgA 1 and 2. And although most of the focus on antibody research looks at the affinity which is controlled by somatic hypermutation, there have been lots of research and studies done in the past that have found a role for the antibody class in dictating the fate and function of a B cell as well. So whether a B cell expresses IgM, IgG, or IgA potentially could have a major role in the differentiation processes that B cell would experience. And after that stage as well, when the B cells are secreting antibody into the serum, there's also important differences in how these classes behave biophysically, and they sort of may target different types of pathogen more efficiently. So IgE is typically an anti-parasitic class of antibody, while IgA is most associated with mucosal tissues. So this is an important part of the repertoire that although we know at the end point how these are functionally important, we don't understand what role, if any, they may play during the process of B cell development.
And also, how should we visualize these different classes? So some of them literally look different. So they form different shapes. Some don't. But what does it mean? How do we determine whether an antibody has a class IgA or IgG or IgM?
So it's encoded by this constant region gene. So antibodies are comprised of the variable regions, which are what encodes the affinity and specificity. And then after that, we have a constant region gene, which is a sort of very structural component, which in the context of a B cell can actually be expressed as a membrane-bound receptor called the B cell receptor, typically enough. And the isotype of that antibody will influence some of those structural components relating to its transmembrane properties in the context of a B cell. So they might have slightly different cytoplasmic tails of the receptor, which could influence how they signal. And I think that's the most important in the context of our study. But outside of the B cell, when they're secreted, they can also multimerize in different ways. So IgM forms quite large, I think, pentamers. IgA is typically a dimer of two IgA molecules. So they sort of do behave quite differently in different contexts.
Okay, so it sounds like you know quite a bit about these B cells and antibodies. What were the things that you didn't know and which prompted you to do this study?
Yeah, it's a really good question. So we do know a lot about B cells. They've been studied for quite a long time, and these germinal centers themselves were first observed histologically over 150 years ago. But they're quite a hard thing to study in the context of human immunology. Mouse models have really taught us a huge amount about these processes. There's a few key aspects that mean that we can't translate findings from mouse directly to humans necessarily all the time, which were some of the major motivations for doing this study. So just for example, the definition of different B cell populations by the cell surface markers is sort of very, in some contexts, very different between mouse and human. So just the B cell identities themselves are not immediately transferable, at least at first glance. But for us, who are really interested in these questions relating to antibody class and function, a major caveat of mouse studies is that they actually have a different number of antibody isotypes than we do. They lack one of the IgA subclasses, for example.
Is that because they lack one of the genes that are needed for that subclass?
Exactly. There's been throughout evolution a duplication event or actually I think it might have been a deletion event. I'm not sure, but there has been throughout evolution a duplication or deletion of one of the genes that has meant that it's no longer there in mouse, and we have it as well. And the other important aspect of it as well, that it's well known that the cytokines that regulate the selection of these different classes differ as well between mouse and humans. So for us who wanted to learn more about the potential importance of these subclasses to B cell biology, a mouse study was not going to cut it. We really had to look in vivo as much as possible. And to do that, we thought that the single cell approaches were the best way to do this.
Right. So you ruled out mouse studies, let's say, because you wanted to study it in humans, but also I think you didn't want to study, for example, B cells that circulate in the blood.
That's right. They're doing a very different thing to this process of the germinal center, which we can only observe within lymphoid tissues. And while there are models for in vitro activation of B cells, if you, for example, harvest B cells from the blood and you can give them the right mix of cytokines, you can approximate some of these processes. We really don't know how close or not this recapitulates the actual events in vivo. And I think the other reason that we wanted to sort of explore single cell methods with this as well was because this is such a dynamic process with cells cycling back and forth from different parts of the tissue, undergoing quite dynamic changes. I think the promise of single cell has always been this revealing unappreciated heterogeneity within systems, and that was something we wanted to explore as well. And we're lucky enough to find some examples of.
Right. So basically, you wanted to look at the B cell development and evolution in their natural habitat.
Exactly. And I think also moving beyond these sort of stereotypical definitions of B cell populations as well. I mentioned before that I'm fairly recently arrived to immunology. One thing that's really hard to get your head around when you first start working in this field, all the different cell surface markers that are used for flow cytometry, and how they are the same between different studies or how they differ very subtly. And I think understanding the relationships between FACS identified populations across many different studies is very challenging. And so in some ways, me coming to this with only a little background allowed me to take a sort of data-driven approach to what are the populations we observe within the data set, and to then try and understand what they are, rather than the other way around, looking for what we expect and then trying to fit that onto the data set.
As you mentioned, these germinal centers where a lot of the action happens, they reside in the secondary lymphoid organs. And so, I guess it's a bit harder to get hold of them in humans than in mice. You can just go around and like slay people and extract their spleens.
Yeah, exactly. This is the challenge. One of the challenges of human immunology, and not quite as easy to get these tissue samples. But the way that we approached it was a fairly classical answer to this question, though, which is to take human tonsils from children who are having routine tonsillectomies. And there are two main reasons for that I can touch on in a moment. But this is a sort of very easily accessible source of human tissue, which is really representative of the immune system, because these tonsils essentially do the same role as the lymph nodes and spleen throughout our body, which are much more difficult to obtain from healthy individuals. And the other advantage of the tonsil system as well is that these are young children, and so their immune systems, I don't know if I'm sure any parents listening will be able to attest, their young children get sick a lot. So their immune systems are working all the time. And although they're not sick necessarily at the time of the procedure, of the surgery, they are processing a lot of new pathogens all the time. So we get a really good active picture of what the immune system is capable of doing, which in other systems, for example, from elderly patients who we might obtain spleen or lymph nodes from, they will typically be much less active and not give us that complete picture that we want to achieve with the single cell profiling.
How hard was it to get hold of these samples? Can you just walk into a clinic and say, give me your tonsils?
Once you have the relationship with the surgical team, that's more or less what happens. So obviously we had to go through ethical, get ethics approval from the relevant bodies here in the UK. But from there, it's quite a simple procedure because this tissue is going to be discarded anyway. They don't require it for any clinical diagnosis or other analysis. So it's literally going to go in the bin after the surgery. So typically it's a very straightforward process to get consent from the parents and the family, and get the tissue from the surgeon. And there's two main reasons that these patients actually are having a tonsillectomy as well, which is kind of relevant. One is that they've had tonsillitis sort of repeatedly within a short period of time. So they're just getting too sick from these repeated infections. And another reason is that they're just obstructive. They obstruct the airway and can cause sleep apnea in these young children. And I think this is an important point just to mention, because we often get asked of how representative is this tissue of the normal immune system?
Exactly.
Are these kids really healthy? What we see is when we have these two different types of patients, we've got an equal number between those two, and we don't see any major differences between them in their gene expression patterns, which is really reassuring to us that this is a representative sample of the adaptive immune system.
But can you tell those two groups apart based on, for example, your single cell sequencing? Did you, like, if you literally color the your UMAP diagram by those batches, do you see a difference?
Yeah, so we did do that, and no, we didn't. We didn't see, for example, any particular cluster or island of cells in the UMAP that were specific to a particular group of patient, which is reassuring.
We already mentioned, or you mentioned that you did a single cell RNA sequencing, and your motivation for that was that you really want to see that heterogeneity and study that heterogeneity of the B cells. But also an interesting aspect of your study was that you use single cell RNA sequencing simultaneously for two purposes. So for gene expression profiling, but also to understand what their B cell receptor sequences look like. So can you talk about this the study design?
Yeah, absolutely. So as I alluded to earlier, some of our major questions that we were interested in asking were about the role of antibody class in B cell fate and function. So do B cells that express a particular type of antibody behave differently to B cells that express a different type of antibody? And to do this, we really needed single cell resolution of both their gene expression and their antibody that they were expressing and making. So we used the 10x genomics single cell platform, which I'm sure a lot of people will be familiar with, but it is basically a droplet based method to encapsulate single cells in these emulsion droplets and then barcode those single cells, barcode the RNA molecules in those single cells with a unique barcode. And then from that we can extract the gene expression information and then separately amplify the antibody genes to examine those sequences as well in a linked but separate assay. So we're getting from each single cell the gene expression information, and for the B cells we're also getting their antibody gene sequence.
You did interesting analyses on this collected data, like phylogenetic analysis or RNA velocity analysis, but simply based on so I guess you build these UMAP diagrams. Maybe you can talk about what sort of relatively simple analysis you did, and whether you gain any insights just from looking at the simple single cell like gene expression data.
Sure. So we spent quite a lot of time working through this data set. Anyone who's worked in single cell genomics of any sort will tell you that now the technology is so simple you can do the experiment in a couple of days and then we'll spend the next 12 months analyzing it. And so there's quite a lot of time working through this and figuring out the right quality control measures to put in place, filtering out using different methods to filter out multiplets, because when we do these droplet-based experiments, you can often get two cells in the same droplet that can appear as one. So we did quite a bit of work looking for those artifacts. But once we'd managed to clean up the data and process it and start exploring it, we found quite a few interesting things. So I guess firstly we found a lot of the things that we expected. We found the naive B cells that I talked about before. We found quite a lot of germinal center B cells, memory B cells, and plasma cells. We also had a lot of information as well from other immune populations in the tonsil, such as the different T cells, which especially T follicular helper cell population, which is really important within the germinal center. And we also found a lot of some myeloid cells as well. But really, most of our work is focused on the B cells, so I'll spend most of the time talking about that.
So was that intentional? Were you selecting? So I assume you did some kind of fax selection before sequencing, and did you intentionally also select T cells, or were they in the sample just by accident?
So we didn't do anything quite as fancy as a fax. We just did a Ficol gradient prep, which is a very standard way to enrich for mononuclear cells, and particularly those from the immune system. So we processed the tissue and we enriched for the immune compartment. So we don't have any endothelial or epithelial populations in our data set, which would be a whole bunch of different questions that would be really interesting in a separate study. But we did want to include these non-B cell populations as well, because they are very important for the processes that the B cells undergo. And some of the analysis that we did in this paper was to look for the co-expression of ligand and receptor pairs between B cells and these other immune cell subsets, to sort of tell us a little bit more about the sorts of communication that might be going on between these different types of cell. But it was, I think, that part of the study, including these T cells, part of a more general, I guess, atlasing approach to this tissue, which had not yet been profiled by anyone else, that we thought it was a useful contribution to make to those studying these populations as well. So
maybe you could explain what this atlasing approach means.
I guess atlasing approach is sequence it all and see, and it'll be useful to someone at some point, I think would be a very crude way to describe that. And I think we're seeing a lot of power to that through studies like the Human Cell Atlas. But we knew, I think there was partly a selfish reason that we wanted to see if there was this potential crosstalk between immune populations, but we also knew it would be valuable to collaborators of ours moving forward as well.
When you try to classify these cells into B cells and T cells and various subtypes of B cells, how, like in practical terms, how easy is that to do? Is there an existing piece of software that will do that for you, or do you have to go to Wikipedia and transcribe like different expressed receptors to analyze it by hand?
That is the second one, is the way that I did it, because when I first generated these data sets, 1 or 2 years ago now, when I first started working on them, these sort of automated classifiers were very much in their infancy. For someone setting out on this path now, they've got so many more options available to them, and then there are there's a lot more data available, and there are a lot more prediction models that can classify the cell types for you. I would say, though, that although I have not used these myself, I think you need to be slightly careful in the fact that if you have novel or unexpected populations in your own data set, how those might be processed by existing annotations would be something that you need to be very careful about. So I would always recommend maybe moving forward a two-prong approach. One, exploring some of these prediction methods to save some of the heavy lifting for you, to do this sort of broad categorization of B cell, T cell, macrophage, endothelial cell, some of that heavy lifting for you, but then to really just explore and subcluster away until you are satisfied that you haven't missed anything that you can't explain. Because it's great to be able to explain what these clusters are, but I think where it gets really exciting is where you find a little population that you don't know what it's called. And that can get a bit laborious after that, of Googling different gene markers and trying to figure out what they are. But I think it's also where this sort of data-driven approach is really best suited and really excels, I think, in we shouldn't try and put too many labels on these things sometimes. And I think this has been a major problem, in my view, in immunology, where any slightly newly classified population by cell surface markers is given a slightly new name. And whether they actually are new or different is, I think debatable. And one of the really interesting questions that I think our study sort of shines a light on in some examples is the difference between cell identity and cell state. You can have a lot of different states for a single cell type, which might appear very differently in these gene expression assays. But how do you approach that, I think, is a slightly philosophical question for people doing these type of work.
Is the distinction that cell identity is non-reversible versus cell state can sort of transition between different states?
That's how I would view it. So you could, I guess, a simple example would be activation of a B cell. You can go from a naive B cell and be activated. I would say you're still a naive B cell. You're still a B cell that hasn't undergone any changes to its antibody genes. It's still antigen inexperienced in terms of its potential function. It may be being exposed to an antigen, but that hasn't had a permanent effect on that B cell's identity, I would argue.
But once you are, if you're a B cell, once you're activated, you can't really go back, can you?
You could.
Oh, you could.
That signal could be taken away, and you could revert to a basal state. I think basal may be a better word than naive in this context. Or you may die. So I guess that's a different question, perhaps. But for example, maybe I can give some more examples of the states that we observed within the memory B cell population, which we focus on in quite some detail on in our paper. We found a subcluster of cells that had a really strong enrichment for the heat shock protein response. So lots of genes linked with that. Now, we can't say absolutely what's going on with these cells, but they're definitely doing something quite distinct from other memory B cells that doesn't appear to be related to their actual identity. They may be undergoing some type of stress in the tissue or during the processing as well. We can't exclude that. And we also identified a subpopulation that had a strong interferon response. So again, this could just be related to the cytokines or other microenvironmental cues that those B cells are experiencing, rather than them necessarily being a distinct and separate lineage.
And while you were classifying, you're giving labels to at least the mainstream subpopulations of B cells. Was there much left? Was there anything that surprised you that you didn't expect to see there?
Yeah, so we found this one population which we've labeled as a pre-germinal center B cell, and these cells firstly were naive in terms of their antibody repertoire, so they were expressing exclusively IgM and IgD, and they had not undergone any somatic hypermutation of their antibody genes. So we were able to say pretty confidently these are a naive population. They did, however, show some evidence of starting to express some genes associated with the germinal center at very low levels, and they lacked some of the key canonical markers, though, of that process as well. So we sort of were getting hints that they might be starting to participate in that response. But that's a difficult thing to test experimentally, so we sort of tried to model that with some computational approaches, and namely RNA velocity. So for those who don't know, RNA velocity is a new method of sort of reconstructing pseudotemporal events within a static snapshot picture that we have from a single cell experiment, because we're only sampling the cells in our population at a single time point. We need to try and reconstruct the potential for cells to move between different states. And RNA velocity has a really, really cool way of approaching this, which is modeling the splicing rates between exons and introns as a pre-mRNA versus mRNA molecules. And that, I think that's a conversation for a whole other podcast for you, Roman. But I think I'll just leave it at that and say that we use this very cool method in the context of our B-cell single-cell datasets to ask the question of whether these pre-germinal center B cells showed any directionality in these splicing dynamics that might suggest whether they were likely to be entering the germinal center. And we found that actually it was a pretty compelling result, that they were, transcriptionally at least, showing a very strong directionality that they were transitioning away from the naive B cell state into the germinal center state. Really sort of, I guess, supporting our hypothesis based purely on the clustering first and foremost. Whereas the RNA velocity is really looking at it at a single cell level, not relying on any of these cluster labels. We can just see for each single cell, is that cell more or less likely to be moving towards other cellular populations in the data set. So we thought that was quite convincing evidence. And we also explored other pseudotemporal methods, and we found the same relationships.
But did you see any evidence that these weren't just a subpopulation of the GCs, of the germinal center cells? These weren't just cells that recently arrived in the germinal center, but this was like a truly discrete, separate subpopulation.
We can't exclude that some of these cells may be present within a germinal center, that they've only just arrived within it. We can't exclude that completely. I think what we can say is that this cluster lacks a lot of the major germinal center marker genes required for localization within the germinal center, and they are known to mark these major cellular populations. And that they've not yet had any change to their antibody genes, and we find no evidence that they have expanded clonally or proliferated yet. We have another piece of evidence that wasn't included in this paper but is now currently under revision elsewhere, which was a collaboration with Omar Bayraktar's group at the Wellcome Sanger Institute in Cambridge, where they've been looking at spatial transcriptomics datasets of the human lymph node, so a similar model system to the tonsil. And we worked with them to generate a reference single cell dataset of all the different immune cell populations that we would expect within the lymph node, based on our tonsil datasets and based on previously published lymph node datasets as well. And so we could look in the spatial assay where pre-germinal center cells are likely to be located. And this is, again, a conversation for a whole other podcast in terms of the methodology behind this. But what we found is that these pre-germinal center state was predicted to fall away from existing germinal centers, supporting the idea that they are most likely in the process of forming new germinal centers rather than joining existing ones.
And you didn't stop at single cell sequencing. You also decided you needed to do bulk RNA sequencing. So what is the advantage of bulk RNA sequencing if you already have a single cell RNA sequencing data set?
Yeah, that's a really good question. So I should clarify, when we say bulk RNA sequencing, what we did specifically was to use the bulk antibody repertoire sequencing. So we were able to sequence the antibody genes of hundreds of thousands of other B cells in bulk, so we don't have single cell resolution, but we can generate a large number of more sequences from the same tonsil samples, and specifically from different populations of B cells within those tonsil samples. So in this case, we don't have quite the same level of resolution or granularity. We had to limit it to naive B cells, germinal center B cells, memory, and plasma cells. But what that really allowed us to do was to more confidently explore these clonal relationships at the single cell level by using the bulk. And I think this can be best explained if we think about this as a sampling problem. So in the context of one human tonsil, there can be anywhere up to or even more than 1 billion B cells present. And on average, our single cell assay, we are sequencing, between 2 to 10,000 B cells from a given sample. So we're not sampling a very large amount of the repertoire at all. So when we have a specific question about the relationships between different B cell clusters in our single cell data, so for example, this pre-germinal center population, we wanted to know, could we identify any relationships between those cells and other types of B cells? And if we only had the single cell data, it would be very difficult, I think, to conclude that no relationships were present, because we've only sampled such a small amount of the repertoire. But now when we can combine the single cell repertoire with the bulk data sets, although we're still never going to get close to the full complement within the tonsil, we can add a lot more confidence to the relationships that we observe. And when we fail to observe a relationship, as we did with the pre-germinal center state, where we didn't see the relationships with any other germinal center B cells or other populations, we can just add a huge amount more confidence to that process.
Interesting. And can you talk a little bit more about how exactly you integrated these two data sets and basically how you went about analyzing this data?
Yeah, so we used a lot of packages from the Incantation suite from Steve Kleinstein's group, and it really allows us to, how should I say, so they're obviously generated through similar but quite different library approaches. So required a little bit of standardization in terms of getting the sequences into the exactly comparable format between the bulk and the single cell assay. But from there, we could combine the antibody sequences into the same data frame and work with them all as one. So we're now looking for each donor patient. We have a combined set of single cell and bulk sequences, and within that we can identify the clonal relationships. To do that, there are a couple of main steps. So I talked at the very beginning of the podcast that we cast our mind back then to these sort of the VDJ recombination events that happen in the bone marrow. And this already generates a huge amount of diversity. So when a single B cell exits the bone marrow, they have a very unique combination of V, D, and J genes. And they've also undergone a very unique recombination event which dictates the junction region of the antibody gene. I won't describe exactly what that is, but this is an important part of this antibody sequence, which is going to be unique to each B cell clonal type. So firstly, to identify these relationships, we can separate or subset out every sequence that has the same V gene, the same J gene, and the same junction sequence. And that means that we can sort of subset down to these are potentially related clonally. And from there, we can use a distance-based metric to look for sequence similarity, to further restrict to B cell sequences that are likely to have been derived from the same clone initially, before expansion in the germinal center.
Did you always know exactly where the sequence begins where it ends? Is it because they're like quite short that they're the transcript can be sequenced in full, or is it because the particular enrichment strategy that you used?
Yes, so we know where our sequence ends because it's an amplicon-based method. So we're using primers to amplify most, but not all, of the antibody genes. So with the part of the genes that we're amplifying, we're getting enough information to identify the variable genes at the 5' end, and enough information to get the constant region gene, or the subclass, at the 3' end. But it's certainly not the full RNA sequence, but it's got all the important information. It's got the V and J gene identities. It's got this junction region, which is where a lot of the important stuff happens, and it's got the constant region. And although the amplification strategies are slightly different between the bulk and the single cell, it's actually quite straightforward to combine the two of them together, because the important information is contained within both assays.
Talk a little bit about how you did the phylogenetic analysis on those sequences.
Yeah, so once we've subset out these B cell sequences based on the V, J, and junction regions, and sort of done this distance-based subsetting even further to identify predicted clones, we're able to use these really nice phylogenetic methods. The one that we used was called Alakazam, which is sort of looks at the mutation changes in the antibody genes to construct these phylogenetic lineage trees and infer the likely events that have happened in that B cell's history based on what's observed in that static snapshot example. It's a very difficult thing to work on, actually, because knowing what your ground truth is almost impossible in this case. It's very hard to do a sanity check on a lot of these phylogenies in terms of which B cell actually came first. Sometimes it will be very clear, because you could have if there's a single mutation that's different between one B cell sequence and another away from the germline sequence, because we know what the germline sequence should be. In that very simple case, we could say which one came first. But there are real limitations around when we get to much larger and more complex situations, which is typically what we observed, where we have many, many, many different mutations, and we're only sampling a very small percentage of these clonal families. So even for the largest clones, we had some examples where we had several hundred sequences within a clonotype. But the majority of expanded clonal events we identify might have 3 to 10 sequences. And these are very heavily mutated. So it is sometimes quite difficult to interpret. So instead, a lot of what we did was really looking for the relationships or the presence within these clonal expansion events between different types of B cells. So within an expanded clone, how often did we observe a germinal center B cell with a plasmablast? How often did we observe germinal center B cell with a memory B cell? And that can tell us something about the relationships between them, given these large caveats around sampling depth and reconstruction of these phylogenies.
What do we mean when we say a clone? Because so there is this activated cell that starts expanding. So we have all of its progeny. We could call that a clone, but also, if I understand correctly, and this is something you explained to me, and I was confused about. I was under the impression that first they mutate and only then they expand. But what you explained to me is that happens iteratively. So they mutate a bit, they expand a bit, and the whole cycle repeats itself. And so at each iteration, you have like a sub clone, right? So can you tell them apart? Can you say, well, this is the ground zero clone, and these are the sub clones?
Theoretically, you could be able to do that if we were able to sequence every single cell in that tonsil. If we could capture the complete repertoire, we might be able to do that. But we can't, unfortunately. But it is a really, really good question about what is a clone. And there's lots of different definitions to that. For me, I define a clone or clonotype. Clonotype might be a more accurate description, actually, of the ideas that I'm discussing. A clonotype would be that B cell that has exited the bone marrow, has undergone that recombination event, and is theoretically at that moment a unique clonotype. From there, it could enter a germinal center reaction and expand and proliferate and generate many, many more subclones, which are still part of that clonotype or clone, to use the broad sense of the word. Now, some of those subclones, those lineages, might be a dead end. They might acquire a harmful mutation in their antibody gene. That means that they can't be selected for positively, and they'll apoptose and die. So that could be a dead end to that lineage. But I think one of the things that we discussed at length, I think, before, was that that founding member of that clone may no longer exist because it has proliferated and changed and may have been selected against because it doesn't have as high affinity as its subclones, as its daughter cells now have. So yeah, there's lots of really interesting ideas around that, but we sort of always need to keep these caveats about sampling static snapshots in time in the back of our heads while we're looking at these questions.
And so, from what I understand, you have you can look at these sequence cells at two levels. So, at a coarse level, you have this just clusters, these clones, or what you roughly identify as clones. But then you can be more detailed in within a clone. You can build a phylogenetic tree. And so, what kind of things that phylogenetic tree allows you to see that just looking at a clone without any relationship doesn't? So I think you mentioned, for example, looking whether like different types or classes coexist in the same clone, but that doesn't require a tree, right? That can be done just looking based on looking at the cluster at a clone.
Yeah, that's right. So I think you can, when you're able to reconstruct a large clone, it can get really interesting. When you can see where these changes in antibody class might be associated with an expansion event, this can really be informative. So often you might see that you have a few members of the clone that are one class, say IgM, and then you detect one clone which is IgG, and then suddenly it's expanded massively, really large. Like, you've detected a lot of IgG subclones from that point. In the context of our study, we didn't explore these sort of ideas very much, partly because of this issue of depth, partly because these kind of questions are most relevant when studying an antigen-specific response. So what you're really looking for in those cases are to identify, if we take coronavirus as an example, if we wanted to see the type of B-cell response to SARS-CoV-2, we would sort out all the SARS-CoV-2 specific B cells and construct these lineages. And you would be able to track through that phylogenetic history the acquisition of different mutations. And then you could go and clone those sequences to test how that affects the affinity of B cells to the coronavirus. And that can tell you a lot about the selection and B cell response. So I just give that as an example of the sort of thing where these phylogenetic approaches are most powerful. It's not something that we explored very much ourselves, though, partly because these kids don't have a specific antigen that they're responding to at that time. When we get this tissue, they're probably responding to 1,000 different things at once, and we can't, with any confidence, predict even what these lineage trees might be linked to.
And we alluded to the question whether multiple classes of B cells can coexist inside a clone, and that basically, if I understand correctly, that boils down to asking whether an activated cell already knows what kind of class it will result into, or it will expand into, because originally it's still a IgM IgD cell, right? But is it the case that once it starts expanding, at some point everything switches to, let's say, IgA or IgE, or is it rather the case that some of its children can become IgA, some can become IgG? I think you tried to answer that question. So how did you investigate that?
Yeah, no. So that's a great question. This was actually relates to one of the cool things that we found about how B cells might be switching their antibodies. So the current paradigm is that you have your IgM B cell, and that is activated and goes into the germinal center. And once it's in the germinal center, it can get the right signals to switch to an isotype. Whether there's any predetermination in that, as you sort of seem to hypothesize in the way you asked the question, I don't know if there's any evidence for that yet, but the current model is that they would switch within the germinal center. What we actually found was in this pre-germinal center population that seems to be committed to forming the germinal center, that these are the cells that seem to be primed to do this process. They seem to be about to undergo that switching event either before or at exactly the same time they form a germinal center reaction, which sort of has important implications to how they behave in the germinal center. That sort of gets away from your question a little bit. So,
and just to make sure I understand your answer that you gave so far, so are you saying, because a germinal center is not formed by a single activated cell, right? There are multiple cells that form a germinal center.
Well, I think it's possible it could come from a single cell, but that early process is not well understood, because that's a very difficult thing to track in vivo, especially in human, yeah.
But at least it can be formed by multiple cells.
I think it's possible to form from either one or multiple cells, yeah.
And is your claim that all of these cells that join together, are you saying they are committed to the same class, or what exactly is your claim?
No, I wouldn't say that. I think there's two scenarios there. If we take the one that there are multiple cells forming a germinal center at the same time, I think if our model is correct and that class switching occurs before the germinal center, each one of those cells would make a decision and switch or not. I don't think they would do it together necessarily. They might coincidentally, because they're receiving similar signals, but I don't think there's any evidence to suggest that they absolutely would do exactly the same thing.
So your claim is that they commit to a switching not much later than forming a germinal center. Is that correct.
That's what our evidence suggests. There, and this is also supported by work in the mouse, where they are able to do, much more fine-grained mechanistic studies of the time course of these events. So some of that's already been published, and some of it will be published soon from other labs. But what we think we've found is the cell state that matches these observations in the mouse. That we found this population in human, where this switching event can happen before a germinal center forms. And then within the germinal center, this doesn't preclude further switching from happening.
So there can be further switching.
There can be further switching.
And including within the same clone?
Within the same clone, exactly. So the way in which that happens, I didn't really describe the mechanism of this class switch combination, but it is essentially, if we imagine the locus of the immunoglobulin gene, we essentially have all these class genes in a linear organization where we have IgM and IgD at the 5' end, and then we have downstream of that all the rest of the IgGs, IgA and IgE in a particular order. When we switch from IgM to one of the other classes, those IgM genes are deleted, so that can't go back. So if we switch from IgM to IgG1, we cut out IgM, we'll also lose IgD, and we'll also lose IgG3, because that happens to be upstream of IgG1. So from that point, a B cell could switch again. It could switch from IgG1 to another class that's downstream. It can't go back because now those genes are gone, but it could switch to IgA1 or IgA2 or something else. And in theory, this could happen multiple times within a single clonotype or clone family. Now, whether this is all happening in a single linear fashion, if you like, where, let's say, a B cell is activated, it becomes a pre-germinal B cell, that primes it to switch, it then forms a germinal center, and then those subsequent switching events occur within that same germinal center, or feasibly that germinal center generates some memory B cells. Those memory B cells exit. They can then be reactivated and form a new germinal center via the pre-germinal center stage, and that's where the class switching occurs. And it could occur many, many times in that fashion.
Well, not that many, right? Because every time you chop off further and further your DNA, so there's...
That's right. And there is definitely a hierarchy in the manner in which you switch from one class to another, one isotype to another. So in some situations, you're probably more likely to do, G1 to G2 than you would be to G1 to A1, for example. I should be very clear that this is very speculative, and we don't have a lot of evidence for actually how this might happen in the context of sequential switching. And it is, again, very difficult to make some of these conclusions from our experiments, because we have basically emulsified, homogenized this tissue into a single cell suspension. So we don't have any information about individual germinal centers. What we kind of would really need to know is to be looking at these at single cell level with the spatial resolution as well, with the antibody genes, to say, right, here's one germinal center that's comprised of clones 1, 2, 3, and 4. Maybe it would only be a single clone. There's some evidence that that's the case. But we could really say, okay, this clone is comprising this entire germinal center, and this other germinal center seems to be related to that. How are those two related? Has there been a class switch event within those germinal centers or before those germinal centers these are sort of, I guess, the dream experiments that you might like to be able to look at.
But still, I think this was a very good explanation, and it makes it clear that these class switching events, they are to be taken seriously. It's not like just a gene expression change that a cell can oscillate between. As you say, it can never go back if it deleted that part of its chromosome. Just to make it really clear for myself, first of all, the claim then that the first class switch event, right, and it now has a weight because, as I said, it's not to be taken lightly. It's the first step out of a possibly finite number of steps, and so that first step is at least sometimes taken before arriving at the germinal center or forming a germinal center.
Yeah, that's currently our model, as well as some other people's model, but it's certainly needs a lot more testing experimentally. But I think it makes a lot of sense given our data set and what other people see in experimental mouse models.
So continuing our discussion about classes of B cell receptors, there is this notion that a class is associated with the specificity of the receptor, and so the direction is interesting. So presumably, once the receptor becomes very specific, maybe it switches, for example, from IgM to IgG, and if it's not very specific, it stays at IgM. But there are also alternative explanations. And what did you find in your study?
Yeah, so this has been a long-standing question in the field, because there've been observations made for quite a long time that if you look at IgM antibodies, these tend to be lower affinity than switched antibodies, so IgG or IgA, which are much higher in their affinity. But we sort of found when we were reading a lot of these old papers using bulk RNA sequencing, they would take IgM B cells and compare them to IgG B cells. But there's a bit of a problem about generalization. So IgM tends to be lower affinity, but you can have very high affinity IgM. And IgG tends to have high affinity, but can also have low affinity as well, depending on what you're looking at. So past studies had sort of done these comparisons of comparing IgM with IgG B cells, seeing if they were different in some functional way. And they would find a lot of differences. But we were sort of wondering, could that be related to their affinity, or is that related to their class? And that's kind of important, because when this class switching occurs, is that dictating their response, or is their response purely dictated by the affinity that they acquire throughout that process? If the response is dictated by that class switching, then that highlights that early pre-germinal center event as a really pivotal moment in a B cell's trajectory.
Maybe you could explain, because it is a bit counterintuitive, because the cell doesn't know in advance what affinity it will be able to acquire. So the natural direction seems to be, well, it mutates. If it achieves a good affinity, it can then switch to IgG, let's say. But how would the opposite direction work mechanistically?
So this kind of comes a little bit around to the temporal events and sort of multiple waves of this process occurring during an infection. And generally it is known that early on in an infection, you will get a large wave of IgM first, before you start to produce this high affinity IgG. So it probably relates to needing some flexibility to provide these different things at different stages. So initially, when you're responding to infection, you want to make a lot of IgM, you want to make a lot of antibody. And if it's not that high affinity, it doesn't matter so much. You just want to be able to make some and start trying to have an effect. In the meantime, you can be forming that high affinity response in the germinal center, which takes a little bit more time, a few days perhaps. And so I guess if you can have some cells that are entering this process that are remaining unswitched, they can go in, do a little bit, but then come out fairly quickly and have a role without acquiring high affinity. And in the meantime, some other cells have switched and they're spending longer in the germinal center, and then they come out maybe slightly later, but they're more efficient and more effective.
Right. So you're saying that those that come out earlier, they don't acquire because they spend less time mutating and evolving, they acquire maybe lesser affinity, and they also don't class switch. And it's the absence of class switching that determines that they are going to exit earlier.
That's what we found evidence for in our gene expression analysis. So we were able to compare with B cells within the germinal center and comparing. We limited our analysis to just high affinity B cells based on their mutation signatures. And when we compared highly mutated or high affinity B cells with expressing IgM and those expressing IgG, we found that those expressing IgG had more expression of receptors associated with remaining in the germinal center. So it seemed like IgM would be more likely to exit and leave the germinal center than the switched B cells would be able to. I think there are other explanations for how this might happen, but I think by controlling for that affinity between the B cells we're doing the analysis from, we can remove that a little bit from the equation, because it certainly is a confounding factor. If you just take all B cells and compare IgM with IgG B cells, you see enormous differences in a whole lot of different genes. But if you take high affinity and low affinity B cells and do a gene expression analysis, you see the same gene expression changes occurring. So that IgM versus IgG comparison is really confounded by high versus low affinity. So one of the points of difference of our analysis has been accounting for that confounding influence and only comparing high affinity with high affinity.
And by affinity, because you can't measure directly affinity, as a proxy, you use just the amount of mutation it acquired.
Exactly, which is unfortunately is all we had available within our own data set. But what we were able to do was to use some experimental data from a mouse study, actually. So this study, what they did, they had a mouse and they could expose it to a very specific model antigen. Hen egg lysosome induces a fantastic immune response, and then they were able to sort for germinal center B cells that had a high affinity or a low affinity to that antigen, and did RNA sequencing to compare the signatures of those B cells. So what I did was I took those gene expression signatures and put them onto our human germinal center B cells, and then compared for B cells with a high level of somatic hypermutation of their antibody genes. Did they have a high signature score for affinity based on these studies? And we saw a pretty good correlation, actually, that supported our use of the mutation level as a proxy for affinity. Of course, in an ideal world, we would now combine these same experiments with labeling for the antigens through a site-seq or similar labeling approach, which is now technologically much more possible than it was 3 years ago, 4 years ago when we started the study.
Is that known, or do you have any? Can you speculate? So, what would be the point? What motivation do B cells have to switch or not switch? So, assuming there is something that primes a B cell to remain longer and mutate more in the germinal center, or remain less and exit earlier, why would it choose one form of the antibody in one case and a different a different class in the other case?
So this probably relates to the microenvironment that event is occurring in, so the cytokines that the B cell is experiencing. So, for example, IgA really dominates class switching in mucosal tissues like the gut and the airway. And this is probably because TGF-beta is one of the main, to my understanding, TGF-beta is important to drive that process, and there's more of it there in those tissues. So I think that's what's partly dictating it, is the context of the tissue.
But that would be the late switching, right? That would be the switching long after it exited the germinal center and arrived at the mucosal.
No, I think that would happen in those tissues. For example, in the gut, there are lymphoid structures.
Okay, so it wouldn't actually leave the gut; it would stay in the gut. So it wouldn't go from the gut to the tonsil.
No. So you can have local immune responses in the gut. There is either through the mesenteric lymph nodes that drain quite a lot with the gut, or with these structures called Peyer's patches, which are essentially germinal center-like organized structures that form within the gut tissue to create these responses.
Okay, but if we talk specifically about exiting early and keeping IgM versus remaining for longer, and keeping IgG, so presumably those cells would be in a similar environment.
Yes. Yeah.
Right. The difference is how much time they spend in the germinal center, and then also the class of their antibodies. So why would there be a correlation between the two?
So the other angle on this relates to the type of B cells that are made by the germinal center as well. So the memory B cells or the plasma cells. And these both arise from the germinal center generally. And in general as well, memory B cells tend to be IgM and plasma cells tend to be IgG or switched, so they're not IgM. So again, the same questions about affinity come up around these two cellular populations. So they also do quite different roles. So plasma cells, their primary role is just to secrete antibody at high amounts. They are terminally differentiated. They can never go back to the germinal center. They are fixed, making that one type of antibody from now on. And they will often go to the bone marrow and reside there, and they'll just be pumping out that antibody for the rest of their life, and they can live for a very long time. Memory B cells, on the other hand, are not doing that. They're just hanging around, ready to be reactivated upon a secondary infection by that same virus or bacteria or whatever it might be. Now, where this could come to answer your question is that these memory B cells can then participate in the germinal center reaction again to improve their affinity. So they already have some level of affinity to the pathogen. But it might be that in a secondary infection case, you need a stronger affinity, you need a better response, or maybe the pathogen has changed, maybe it's evolved, and now your initial recognition is not quite as good, and you want to improve on that. So that memory B cell will be activated and enter the germinal center reaction again to acquire more affinity. So obviously that's a point at which it could undergo class switching to go from IgM to IgG. Now, if we were to think about an IgM versus IgG memory B cell, an IgG cell that's already switched has less room to move. It has already cut out a couple of its options for the type of antibody that it could make. So if it switched to IgG1, it might have only lost IgG3 that it can now produce. But in some context, IgG3 could be really useful. Maybe for that type of pathogen, that's a more effective response. If it had switched further to IgG2, now it's lost G1, G3, and A1. If it's gone all the way to A2, it can't switch at all. It's fixed. It can't do anything more in that area to improve itself. So actually, by biasing memory B cells towards that IgM state, you probably allow more flexibility in that secondary response, which is probably what you want. A lot of that is very speculative, I should say, but we do find some evidence in our analysis that IgM B cells in the germinal center are less likely to express plasmablast-specific transcription factors, so would be less likely to differentiate into that form and remain as memory B cells. And we also saw that when we had a deep dive into memory B cells, and we could identify the pre-germinal center state of the memory B cells that seemed to be getting ready for this secondary activation, that the IgM cells were more likely to be primed to undergo that switching event as part of that response.
It's a very good point. It's a good point. And did you also discover anything interesting about subclasses? So not just IgG versus IgM, but IgG1 versus IgG3, as you mentioned.
Yeah, we looked. We really looked quite a lot to identify if there were specific subclass-specific effects, and we didn't find any really convincing evidence that there was. There have been studies published in the past that have identified some differences, for example, between IgG4 and IgG1 B cells. We didn't get any IgG4 B cells in our samples because they're very, very rare, but amongst all the other subclasses, there were very few significant effects.
And effects in terms of gene expression, or effects in terms of specificity?
Gene expression. You do see one interesting feature about the B cell repertoire relating to specificity and affinity is that when you move through the locus, because there's this sequential switching that can happen where you go from IgM to one to another, as you move through that locus from 5' to 3', you see increased affinity because the B cells that have switched to the very terminal isotype have likely gone through many, many more rounds of selection than those that have just undergone to the first one. So there are differences with that aspect, for sure. But and we also looked between IgG B cells and IgA B cells. As I've talked a little bit about, there are differences in their distribution throughout the body, but we found very few differences between them in the tonsil. One interesting example, though, was the gene CLEC16A, which has been linked with a IgA-specific immunodeficiency in human patients, and we found that was more expressed significantly in IgA-specific B cells in the germinal center, which is a really interesting idea to follow up on, I think.
By the way, while you were talking, it occurred to me, do we know if when doing class switching, when chopping out those pieces of DNA, maybe it always does it one by one, or can it jump and can it cut multiple segments at the same time?
Yes, you can jump multiple genes at once, and often that's what will happen. It is a difficult thing to quantify how much either of these processes do or don't occur in a given response, but there is evidence for all scenarios on that front. You can, I say that, I'm pretty sure that there's evidence that you can switch to the very last isotype directly to go from IgM to IgA2, but I suspect that it is very rare, and there is definitely evidence that the distance, as well as many other factors to do with the gene regulation at that site, will influence it. Because class switching to which to different isotypes is dictated by this non-coding transcription that occurs from these intergenic promoters at the locus. So each isotype has a intergenic promoter that is transcribed, and when it's transcribed, it sort of opens up. The model is it opens up the chromatin around that site and allows the machinery to get in there and do the recombination at these particular recombination signal sites. But it's it's an amazing locus, actually. It's very cool.
Are there any other either findings or just interesting aspects to your paper that you'd like to discuss?
I think we had lots of too many things to include in this paper and keep a cohesive narrative. I think this is probably more of a general comment than anything about the science, but I think it's a real challenge with this type of work to tell the story in a way that the journals require you to do it, and at the same time present all of the data at once. So we've ended up with so many different little tidbits of information and ideas that emerge from these large data sets that we could spend a lot of time just going on and on and on about.
Anything you could share without a fear of being scooped?
Well, there was one there's one interesting population that we don't know what they're doing, and if anyone wants to look into them further, very much welcome it. So we found within the germinal center a population of B cells that we labeled FcRl3 high germinal center B cells, and they were characterized by the expression of this FcRl3 and FcRl2, which are these surface ligand receptors, and are associated with inhibited B cell receptor signaling, so the inhibited antibody receptor signaling, which is a little counterintuitive for in the germinal center, because you must require active signaling to remain there.
So signaling meaning that when a receptor finds its antigen, it calls back to the cell and reports about that.
Exactly. And we found that this population also expressed a lot of other receptors, genes associated with inhibitory signaling of the B cell receptor. But conversely, these cells had very high mutation levels, so quite possibly high affinity to their antigen. They were part of very large clonal expansion events, so they're not a dying dead end of a clonotype. But perhaps also conversely, they're almost exclusively IgM. They're unswitched. And we were able to validate by flow cytometry that the expression of FcRl3 on the surface of these B cells was much more associated with unswitched IgM B cells. So this is a really interesting population that we refrained from giving them any descriptive name because we ultimately don't know what they're doing or what they might be doing. But we did also identify a similar state of memory B cell. So this is, again, back to that question of identity versus cell state. And this seems to be a cellular state that can occur across different B cell lineages. What potential functional importance that has or not, we really don't know. We've got a few hypotheses, but we're sort of looking more into that at the moment.
Well, Hamish, thanks for your time, and thanks for educating us about immunology and about your paper.
Oh, thanks so much. I hope you know a little bit more about B cells, understand some of it a bit better, but I really appreciate the chance to share my research with you and your listeners.